Dear students, Assalamu alaikum. Today I will deliver a lesson for class 12th subject zoology. My today's topic is crossing over and its importance. Now, crossing over is actually the mutual exchange of segments between the non-sister chromatids of homologous chromosomes. As you can see in this sketch, these are the two homologous chromosomes, and each chromosome has actually two chromatids. This pairing of homologous chromosomes actually occurs during the meiotic division at zygotine stage of prophase 1. So during this pairing, actually these are called as bivalents or tetrads, because actually in a tetrad or a bivalent there are two homologous chromosomes and each chromosome has two chromatids, and in total a bivalent has 4 chromatids. Now you can see in this case, this is one of the homologous chromosome with 2 chromatids designated as 1 and 2, and another homologous chromosome having the 2 chromatids designated as number 3 and 4. Now these chromatids of chromosome A, that is chromatid 1 and chromatid 2, are the sister chromatids because they belong to the same chromosome. Similarly, the chromatid number 3 and chromatid number 4 belong to the chromosome B and are also the sister chromatids of this chromosome B. Actually, during crossing over, the exchange of segments or exchange of genes takes place between non-sister chromatids. As you can see, the non-sister chromatids in this case will be 2 and 3 non-sister chromatids. Actually, the exchange of segments occurs between non-sister chromatids, and this exchange of segments, or we call it as crossing over, actually takes place during the meiotic cell division at packaging stage of prophase 1. Now, crossing over actually takes place by breaking and reunion of these chromosomal segments. Now, breaking of these segments, also known as nicking, actually occurs with the help of an enzyme known as endonuclease. And then these broken segments get healed up, or the reunion of these broken segments takes place, which is also known as annealing. And this annealing is actually done by another enzyme known as ligase. Therefore, so you must remember, endonuclease and ligase are the two important enzymes needed for breaking and reunion of these broken chromosomal segments. Now, this type of exchange, as you can see here, the exchange of these non-sister segments of these chromatids, this type of exchange of segments brings about the chromosomal recombination. For instance, you can see this is the chromosome, one of the homologous chromosomes is another homologous chromosome. Now at the packaging stage of prophase 1 of meiosis, this exchange of segments takes place, and the site at which these chromosomes actually cross each other is known as chiasma. Now, whole of this process results in the formation of recombinants. As you can see, these are the recombinant, these chromosomes, in which this part of the segment is exchanged with another part of the homologous part of this chromosome. Now this is the crossing over, and there are various factors which affect the process of this crossing over. Now we will understand about the factors affecting this process of crossing over. The main factors that affect the crossing over are distance, age, sex, and exposure to radiation and temperature. So far the effect of distance is concerned, the frequency of crossing over increases with increase in the distance between two genes in a chromosome or in a chromatid. Similarly, age is another factor which affects the crossing over. In fact, crossing over, the frequency of crossing over decreases with the increase in age. Sex also plays an important role and is a factor that affects the crossing over, as is seen in various experiments done on Drosophila and rats, and it has been seen that males have low frequency of crossing over than females. Now, radiation and temperature are another two important factors which affect the crossing over. For example, exposure to radiations like X-rays, ultraviolet radiations, and also variations in temperature increases the frequency of crossing over. Now, this was about the factors that affect the process of crossing over. Now we will lastly discuss about the importance of crossing over. The crossing over has many importance or significances, but some of the most important significances are: number one, that crossing over increases variability, and this variability actually plays an important role in the process of evolution. This is the most important significance of this crossing over. Another importance is that crossing over results in the formation of new genetic combinations, which are known as recombinants. As is seen in this case, these are the recombinant chromosomes, and the useful recombinants produced by the crossing over are selected to produce new varieties of plants as well as of animals. For instance, the famous grain revolution, which you may have heard about, and also the white revolution, have been actually achieved by the selection of recombinants which are obtained through crossing over. Now this is all about the crossing over and various factors affecting the crossing over and the importance of crossing over. Goodbye.